This video is brought to you by Global Men's Wellness, your 24/7 AI health expert. No copays, waiting rooms, or awkward conversations. Just real answers whenever you need them. Visit globalmenswellness.com to try it free and get your own on-demand concierge-level wellness expert today.
Welcome to the 21st Century Man podcast. We're doing our deep dive series, looking at each chapter of the 21st Century Man, the guide, edited by Dr. Judson Brandeis.
And today, we're tackling Chapter 84. It's titled Stem Cell Medicine, What Does the Future Hold? The author is Dr. Jeffrey Petrillo, who's an orthopedic surgeon and also a doctor of regenerative medicine.
Yeah, so our goal here really is to unpack this chapter using the source material, kind of cut through the hype.
Right. Separate the science from maybe the science fiction, give you a clear picture of this field because it's incredibly promising, but there's also a lot of noise out there.
Definitely.
Yeah.
And the editor, Dr. Brandeis, he says that right at the start. Stem cells, huge promise for curing diseases, maybe reversing aging. But, and this is the big but, it also creates this space for, well, fraud, false promises, unrealistic expectations.
Yeah, it's a tricky landscape, and that's exactly why the FDA has gotten involved and put pretty strict limits on most stem cell products.
Important clarification, though. The source notes that PRP, platelet-rich plasma, isn't included in those specific restrictions. We'll get into that later.
Good point. But the chapter itself kicks off with a really powerful story.
It does. About Dr. Gregory Giamanco, a chief resident.
Right, and this young patient, 17 years old, paralyzed from an ATV crash, spinal fracture.
Yeah, really tough situation. And then it pivots to Giamanco's research lab. They called it the Barn.
Yep, Building 14A, part of the mammalian spinal cord research program. Pretty high-tech stuff, actually.
And the research described as, well, intense. Severing animal spinal cords, injecting stem cells.
And trying to see if they can restore function, it really grabs you.
It does. It's designed to make you pause and ask, is this real science happening now, or is it still kind of sci-fi? Sets the stage perfectly.
And they connect that idea to Margaret Atwood, interestingly. How back in 2003, she wrote about things like transgenic pigs for organ transplants as far-fetched science fiction.
And the source points out, well, today, it's not so much fiction anymore.
We're actually working on these knockout pigs, genetically engineered to grow tissues, organs.
That humans could potentially use.
Oh.
Without rejection.
That's the goal. Avoid rejection, even fight off microbes. They're even splicing in genes to make things mature faster. It shows how quickly things are moving.
Okay, so let's pull back a bit. The chapter talks about this broader field, regenerative medicine. What's the basic idea there?
Well, regenerative medicine is all about trying to repair, restore, or even replace damaged tissues and organs.
And stem cells are key to that, helping with aging, pain, that kind of thing.
Exactly, because stem cells have this amazing ability to become different types of cells. The hope is they can help us, maybe age better with less pain, less degeneration.
Which leads to the basic question, what are stem cells? The chapter gives the short story.
Yeah, basically they're specialized human cells that can differentiate meaning, turn into many different cell types. Different cell lines.
And that's the potential, right? Replacing cells that are damaged or defective. For injuries, Parkinson's, heart disease, diabetes.
That's the promise. And the chapter breaks them down into two main types, embryonic stem cells and adult stem cells, sometimes called tissue stem cells.
Okay, now this hierarchy thing is interesting. How they classify them based on potential. Let's start at the top. Totipotent.
Right. Think of totipotent as the master cell. It can become any cell type in the animal, plus the supporting tissues like the placenta.
So the very first cell, the zygote after fertilization, is totipotent.
Completely. And the source uses that powerful image. That one cell makes everything cartilage, hair, heart, even, the penis example they use, every single tissue.
Imagine tapping into that power later in life. The healing potential.
It's mind-boggling. So that zygote divides and the cells start to specialize a bit. They form three layers, ectoderm, mesoderm, endoderm, which are blueprints for different systems.
And at that point, they're no longer totipotent, but pluripotent.
Exactly. Pluripotent cells can still become any of the body's 220 or so tissue types, but they can't make the placenta.
So embryonic cells after those first few divisions.
That's them. And the source highlights these as holding the most promise for regenerative medicine because they're so versatile, but they also come with more ethical and regulatory complexity.
Okay, and then there's multipotent.
These are more limited again. They can develop into more than one cell type, but within a narrower range. Adult stem cells, like from fat or bone marrow, fit here.
And also the cells from placental sources, like the cord, Wharton's jelly.
Right. Those also fall under the multipotent category. They're not quite as flexible as pluripotent, but still very useful.
The chapter also hits on a really important point. Where do we get these cells? Ethically, safely, and, in large enough quantities.
Yeah, and it points to the full-term human placenta, often discarded after C-sections, as a really promising source.
What's that?
Well, it's available in huge quantities. There aren't really ethical issues like with embryonic cells, and it's easy to get. Plus, placental cells seem capable of differentiating toward all those 3 basic tissue layers.
So they might regenerate lots of different tissues.
Potentially, yes. They might hold on to some of that early growth potential without harming an embryo. It's a really interesting area.
Okay, so based on all that, if you're listening and dealing with an injury or a disease or just feeling the effects of aging, what does the source say is actually available now? What are men asking about?
Good question. The chapter looks at current options. It starts with something many people have heard of: platelet-rich plasma, or PRP.
Right, been around maybe 20 years, used to try and slow down aging effects.
Exactly. You take someone's own blood, spin it down, concentrate the platelets and the growth factors they release.
The author, Dr. Petrillo, mentions he started using it way back in 2002. It started in dentistry.
Yeah, then orthopedics, now wound care, aesthetics, sexual health. It's used quite widely. And because it's your own blood, it's autologous. Donor and recipient are the same.
Safe, generally, but the chapter points out a catch with autologous PRP, doesn't it?
It does. The quality really varies. PRP from a fit 20-year-old is just different than from a 65-year-old with diabetes, for instance.
So researchers are looking at ways to boost it for older people, like exercise before the blood draw, or washing the platelets.
Yeah, or freezing the plasma, adding other factors, trying to make it more potent, more consistent.
And the chapter kind of frames stem cells as the next level up, the penultimate destination beyond PRP.
Right, and it zooms in on the placenta again as a key source researchers explored next.
Makes sense. Amniotic fluid develops a whole human life, must be packed with growth factors.
Totally. It contains multipotent, maybe even some pluripotent stem cells. And this is where tissue engineering, maybe growing new tissues, starts to feel less like that Atwood sci-fi.
So they look deeper into the actual placental tissue, the placenta itself, the umbilical cord stuff, usually thrown away.
And found they were really rich sources of stem cells with unique properties compared to adult stem cells. The chapter then lists some products derived from these.
Like Wharton's jelly.
Yeah, that gelatin-like stuff in the umbilical cord. Rich in hyaluronic acid, good for joints, and it contains mesenchymal stem cells, MSCs, first described ages ago, 1656.
What's really striking is the concentration difference the source mentions.
Huge difference. Wharton's jelly might have 1 stem cell for every 300 other cells, maybe up to 1 in 1,700. Compare that to adult bone marrow in a 60-year-old, which is more like 1 in 2 million.
Wow. That's a massive difference in starting material.
It really is. And these Wharton's jelly MSCs have big advantages listed in the source. They come from healthy donors, they're immune privileged.
Meaning no immune reaction, no tissue matching needed.
Generally, yes. They can kind of fly under the immune system's radar. They're also non-tumorigenic, according to the source. And they're young, so they divide super fast.
Like one cell could make 1 billion in a month, compared to just a few hundred from an older cell.
That's the comparison the chapter makes. Plus, they can differentiate into bone, cartilage, etc., and release way more bioactive molecules, like 300 types, versus maybe 100, 200 from older cells, helping with healing, inflammation, regeneration.
Okay, what about using live placental stem cells intravenously? The chapter mentions that too.
Yeah. Potential IV uses suggested include reducing systemic inflammation in autoimmune conditions, maybe helping with early Parkinson's or Alzheimer's, general anti-aging effects.
Again, that immune privilege status is key.
Absolutely. No HLA markers means no rejection, theoretically. And because they're young, the source says their therapeutic activity is just stronger, more growth factors, faster differentiation, slower aging, better anti-inflammatory and nerve protective effects compared to older adult MSCs.
Now, exosomes. This sounds really new and interesting.
It is. The source describes them as potent little messengers derived from placental fluids, kind of like the beneficial cargo from placental stem cells, but without the actual cell structure.
So catalysts for health, altering inflammation, tissue repair, nerve communication.
All that, plus mitochondrial health, immune regulation. The idea is they can target almost any damaged cell joint, heart, brain. Very new use, but promising.
And a key benefit mentioned is lower risk compared to injecting whole cells.
Right. The source brings up the theoretical risk of chimera formation with whole cell transplants donor DNA mixing with yours, like how a bone marrow transplant shows up on a 23andMe test. Exosomes, lacking that core cellular machinery and DNA, seem to avoid that specific risk.
How do they work then if they're not cells?
Cell-to-cell communication. They use specific molecules, like peptides, to send messages to your own stem cells, telling them to wake up and start repairing tissue. They're like signals, not the building blocks themselves.
Got it. And the last placental source mentioned is amniotic tissue.
Yeah, another injectable biologic, this time from the amniotic membrane lining, can be used with PRP or alone. It's called provascular, helps new blood vessels form, reduces inflammation.
And it signals the body's own stem cells to come to the area.
That's the idea. Contains proteins that act like homing signals for your own regenerative cells.
And these products come from amniotic fluid aspiration or processing the lining after dissections.
Both methods are used. They're rich in growth factors, anti-inflammatory cytokines.
But there's some debate here about live cells in the final product.
Yeah, the chapter notes that. A 2019 study found live cells in fresh amniotic fluid, but maybe not so much in the processed off-the-shelf product. So still an area being figured out, a work in progress, as the source says.
Okay, let's switch back to autologous therapy, where you're your own donor. We covered PRP from blood already.
Then there's bone marrow concentrate, or BMAC. Your bone marrow makes blood cells, but it also has MSCs that can create bone, cartilage, fat, blood vessels.
And they get this by aspirating bone marrow, usually from the hip bone, under sedation.
Right. Minimally invasive. Then they process it, centrifuge it like PRP to concentrate those stem cells.
But the big issue here, the source emphasizes, is age again.
Definitely. Just like with PRP, but even more pronounced, the number of viable stem cells in your bone marrow drops massively as you get older. Processing methods also vary, affecting quality.
Those numbers again: birth, 1 in 1,500, age 60, maybe 1 in 2 million.
Exactly. That stark decline is a huge reason why researchers are so focused on those younger, more plentiful placental sources we talked about.
The final autologous source is micronized fat, MSAT. Fat tissue has MSCs too.
It does. And it's been used successfully in cosmetic surgery for a while, facelifts, sexual health enhancements. That success got orthopedics interested for things like joint injections.
Wasn't there some controversy with a fat technique? Stromal vascular fraction.
Yeah, that used enzymes to release the stem cells.
Oh.
The FDA ruled against it under Section 127, saying the enzyme manipulation turned it into a drug needing full drug approval.
But micronized fat aspirate, MFAT, is still allowed, just mechanically breaking down the fat.
Correct. Though the source mentions there's still some debate about whether the stem cells are truly released or if the benefit comes from the whole microfragmented tissue environment.
But there's positive research, like that Emory study on knee osteoarthritis.
Yeah, the 2019 study mentioned found MFAT injections gave significant improvements in pain and function, pretty comparable to BMAC results. And the fat aspiration procedure was generally less painful than the bone marrow one. Both showed benefits.
So all this stuff is becoming more available, more sought after. The future is now, as the chapter says, leads to stem cell tourism.
Right. People seeking treatments here and abroad. Which brings us to some really crucial practical advice the source offers for you, the listener.
This is key. First off, these aren't simple procedures. You need someone qualified.
Absolutely. Find a provider with specific training, experience, and a documented track record. Don't be afraid to ask for credentials and proof.
And shop around, right? Don't just go with the first place you find.
Definitely. Compare providers, ask questions, see who feels right for your needs.
And that point about cost.
Yeah.
Cheapest is rarely best here.
Very rarely. Quality in collection, processing, and the expertise of the provider all cost money. Be wary of deals that seem too good to be true.
So if the price feels way off too high or too low, or if you just get a bad vibe,
trust your gut. The source says it's totally okay to walk away, do more digging, maybe get a second opinion. Your health is paramount.
Love the Mae West quote they included. You are never too old to become younger. Captures the hope, doesn't it?
It does, but then the chapter pivots to the essential regulatory side of the FDA. Super important given the potential for scams.
So the FDA does regulate these?
Yes. Standard uses like bone marrow transplants for cancer are approved, but for these newer investigational uses, joints, anti-aging, neuro stuff, they require a formal process.
The IND. investigational new drug application.
Right. That involves submitting detailed plans, manufacturing info, animal data, and then conducting proper clinical trials to prove safety and effectiveness before it can be widely marketed.
So watch out for clinics claiming they're doing trials without an IND or saying FDA approval isn't needed.
Big red flag. The source is very clear. Treatments not under an FDA-allowed IND haven't been vetted by the FDA for safety or whether they even work.
And the FDA is worried about vulnerable patients being exploited.
Exactly. They're trying to crack down on illegal, potentially harmful treatments while still encouraging legitimate research.
So the absolute bottom line for the listener is,
Make sure any stem cell treatment you consider is either fully FDA approved or being studied under an official FDA-allowed IND. Anything else is unproven and potentially risky. Don't believe the hype from clinics that skirt the rules.
okay, wrapping up. The chapter really emphasizes the amazing potential here, but also how understanding the science makes you a smarter consumer. Yeah.
Totally. Knowing the difference between totipotent, total potential zygote, pluripotent, all tissues, no placenta, high promise, and multipotent, more limited adult placental cells, helps you understand what's being offered.
And that final warning bears repeating. Buyer beware. Research the doctor. Get a second opinion if you need to. Trust your instincts about the provider and the price.
Absolutely. It's a complex field, and this chapter by Dr. Petrillo in Dr. Brandeis's book, The 21st Century Man, does a really great job laying out the science and the practical realities. Very helpful for separating fact from fiction.
We agree. It's an excellent resource. We definitely encourage you to check out the full book, The 21st Century Man, for more on this and tons of other topics.
You know, thinking about it all from those sci-fi sounding pigs to the exosomes acting like cellular messengers, it really is amazing how fast this field is moving. The potential feels huge, but harnessing it safely and ethically, especially with these young, powerful cells, that's the ongoing challenge, isn't it?
It really is. Definitely leaves you with a lot to think about. Well, thank you for joining us on this deep dive into Chapter 84.
Hope it was informative and gave you some clarity.
Join us next time on the 21st Century Man podcast for our deep dive into Chapter 85, when a penile implant is the best option. We'll see you then.
Take control of your health anytime, anywhere. Visit globalmenswellness.com and start your free trial today. Get expert-level answers in seconds, not weeks, without the cost, the wait, or the awkward conversations. Real support on your terms. Visit globalmenswellness.com now.